Hello, I am John Wojon, otherwise known as Michael. I'm the developer of G2PMineR, and I have created this video to explain how to use the EasyG2PMineR pipeline, which is a simplified classroom-ready version of G2PMineR that can be run within a single class period. So the first thing that students need to do is they need to open the EasyG2PMineR_pipeline.r file. You do this just by double clicking on it like any other file, and it should open up in RStudio as it is shown here. RStudio is a programming environment where you can interact with the R programming language. So the first thing that we're going to need to do is we're going to need to install G2PMineR. We do this by just going to this section here. Using our mouse, we highlight lines 7 to 10, and then we hit this run button. Highlighting and hitting this run button is how you run specific parts of an R script. Now it might come up with a message like this. What you need to do is you just hit yes. There we go. So here we can see that now everything is installed, and you can confirm that by making sure that there is no more output being written to the console. The next thing we're going to do is we're going to confirm where our output will be written. We do this by highlighting this get wd function here, and then of course hitting running. You can see that it's going to be writing it to the desktop, which is where this EasyG2PMineR pipeline file is located on my computer. So the next part is we're going to actually do the analysis. You can see that we have the EasyG2PMineR function and 4 arguments to the function. These arguments are what the function eats and uses to create the output. First, we have a query. The query here is what G2PMineR is going to be using to look for abstracts. So here we can see we have 3 parts to it. We have this first part, the second part, and this third part. The first part is your organism of interest. I recommend making it broad, like plants, animals, fungi, something like that. Our second part here is our phenotype, in this case, salt tolerance. Could be heat tolerance, could be growth regulation, could be anything that you want. And then this last part, you're never going to change. It's just telling the search algorithms that you want things that have genetics in them. Now here we have our kingdom. Our kingdom is either going to be P for plantae, A for animalia, or F for fungi. Our seed is going to be the way that our EasyG2PMineR analysis is made reproducible, so that the random parts of G2PMineR are really pseudo-random and can be redone with the same seed. Now, it's important for every group to have a different seed. How many abstracts are going to be taken as a random sample from all of the abstracts that are found using the query? And then this is the function itself. So to run this, we are just going to highlight this whole thing, and then we are going to hit run. It's going to produce a whole lot of output. So you'll know when the analysis is finished, when there's no more output being produced on the console. In the next video, I'm going to show you guys how to interpret the results of the EasyG2PMineR function. So to interpret the EasyG2PMineR pipeline results, what you do is you navigate back to the output directory, in this case our desktop, then we look and we will notice there are two new files, interpretable results.pdf and g2pdata.csv. Interpretable results.pdf, if you open that, you'll see that it has 3 pages. You have this first page, which is this computer written output here that tells you your query, the date it was produced, and the results of each of the species, genes, phenotype searches, the percent of abstracts that had matches in each of those categories. And then the second part here is on the third page. This is the bipartite G2P, genotype to phenotype output graph. So up here on this upper level, you have the phenotypes represented by these black rectangles. The thicker the rectangle, the more abstracts had those phenotypes within them. Now, in here we have the genes, same thing with the thickness of the rectangles down there. Connecting the genes and the phenotypes, we have these gray lines. The thicker these lines are, the more connections there are. So the more abstracts have both this gene and this phenotype in their texts. So you can see, for example, if we go to the word drought, you can see that drought is connected to NAL1, CITRX, CYSC, as well as several others. Now, once you have your hypothesized genes of interest picked out, what you do then is you go to your G2P_data.csv file. You open that up into your favorite spreadsheet editor. In this case, we're going to be using Excel. You can see it has the list of genes here, and then we have the family, and then the ontology if available. So going back to here, let's say we're looking at NAL1. If we go to here and we look at NAL1, we have it here. You can see that this is involved with internode patterning, leaf vascular tissue pattern formation, leaf development and vascular tissue formation. Now, we could say that this is a possible gene involved with salt tolerance because it is affecting the vascular tissue of the plants. And so I would like you guys to do that with all of the genes here. That way you can form your hypotheses in an informed way. That is how you interpret the EasyG2PMineR results. I hope you enjoyed performing this exercise, and I hope that you have a nice day. Bye-bye.